Hi there, Mark here again, and this is the final step in our paper modeling activity. This is the final section. In this part, you will essentially be creating models involved in stopping the coronavirus. So we've covered its overall shape, we've covered how it binds to a host cell, and we've covered kind of what happens once it injects its RNA into that host cell. But a lot of people right now across the world are very focused on how we stop that from happening. And the two main ways are antibodies. So antibodies essentially are our natural immune system that learns the shape of a coronavirus and then is able to bind to it and identify it and make sure it gets destroyed. It's also part of a vaccine story. Essentially, you're, with a vaccine, tricking your body into creating these antibodies so that they can attack the coronavirus if you should get infected with it. And then the other way scientists are exploring to stop the coronavirus is small drug inhibitors, essentially ways of stopping the functionality of the virus once it's already gotten into someone. Those include things like ACE2 decoys. So instead of an actual ACE2 receptor, it's a small fragment that looks like it that'll bind to all the spike proteins on a coronavirus, preventing them from actually binding to the real ACE2 receptors on our host cells. Or small molecules that may bind to, for example, that protease protein. If you remember, that's the one that cut that polyprotein up into individual proteins, allowing it to make more virus particles. And so by stopping that protease with a small drug inhibitor, you may prevent it from being able to cut up that polyprotein. That's essentially what we're going to be modeling in this section of the paper activity. Again, like the other models in this paper modeling series, this is available as a color PDF, a black and white PDF, as well as a drawing guide in case you don't have access to a printer at all. And just like the others, you start by cutting along any of the dashed or dotted lines. Once you've finished cutting, what you're left with are three little piles. One is ACE decoy pieces, one are antibody pieces, and one are this protease with a inhibitor on it. All of these are involved in different techniques being researched to stop or inhibit the coronavirus. The first one we'll look at is this little green guy. If you remember, on our host cell membrane, we have the ACE2 receptor. It's what the spike protein of the coronavirus binds to when it's trying to invade a host cell. So what scientists are exploring is taking just a fragment of that ACE2 receptor, just a piece that binds to the binding domain of the spike protein. And so their hope is that if these little green ACE2 decoys or ACE2 mimics can bind to the spike proteins on the coronavirus, it will prevent it from being able to bind to the real ACE2 receptors that are on our cells. So you can represent this simply by taking some of these cut out green ACE2 fragments and taping them to a few of our spike proteins. In theory, these spike proteins that have now bound to this ACE mimic would not be able to bind to the actual ACE receptor on our cells, thereby preventing the coronavirus from attaching and gaining entry into our cells. The next one is an antibody. Antibodies are a natural part of our immune system that we develop when we've been exposed to either something like a virus, a foreign protein, or if we've been exposed to a vaccine, which has essentially fragments of the proteins that are on a virus. And these antibodies have a binding domain right on the ends here. And those can, similar to the ACE2 mimic, attach to our spike proteins. And it has a similar result to our ACE2 decoy proteins. Essentially, we've blocked these spike proteins. They can no longer attach, and the antibodies signal this particle for destruction. The last thing we have here is essentially the protease that we were working with earlier. So if you remember, our viral genome that's injected into the host cell translates to a number of different proteins that are all part of one long polyprotein initially, until our protease comes along and cuts them separate from each other, allowing more viruses to form. One of the techniques researchers are looking at is inhibiting this protease. So instead of having these little star representations implying that it is able to cut, there's a small molecule bound in each of those cut sites, meaning this can't go and successfully cut the polyprotein into individual proteins. This prevents the virus from forming new viruses and stops the infection. By the end of this activity, you will have your coronavirus with its spike proteins, the ACE2 receptor on the host cell membrane, the genetic payload that you know gets translated and then cut into individual proteins, as well as some antibodies and some small drugs that are able to inhibit or stop the coronavirus from doing this whole process. Hopefully these paper models will help you better understand this entire coronavirus story, and hopefully they can be used in conjunction with some of the other videos on this webpage. Good luck!